Good afternoon, everyone. I'm Shoji Takeuche from the University of Tokyo, and thank you very much for the kind invitations. And today I'd like to talk about the biohybrid devices that we are now working on. And in this slide, this cartoon shows one of our research goals, which shows the kind of humanoids that's combined with the biological materials. And it looks like a human beings, but having the very super sensors on the surface, and also they have the self-healing or self-reproducing abilities, and they work as they work using the chemical energies like biological materials. That is kind of our research directions, and why we are now doing so. So as you may know, the human beings have created the wonderful systems like humanoids, smartphone, or self-driving cars. But yet, we still cannot make the very wonderful functions that the biological systems have in nature. For example, the molecular detections, the highly sensitive molecular detection like a dog nose, or self-healing or self-reproducing abilities in our cellular systems, and highly energy efficient motions or processing like in muscles or brains. Those kind of the functions is very attractive as a researcher in the engineering field, but very hard or very difficult to create by using the artificial systems from scratch. So if it's so difficult to create a whole biological system from scratch, why not using the biological materials directly combined with the artificial systems? That is a kind of our concept of the biohybrid systems that we are now working on. So this is our previous work. World first the biohybrid robot fingers that consists of the skeletal muscle tissues with the finger bone-like structures. So when we apply the voltage between the muscles, which is made of the tissue engineered muscles, the muscle make a contraction motions and they pulls the fingers so that the finger can move back and forth. So it's total robotic motions. And how we formed this kind of the muscles. So we actually use the bottom-up tissue engineering approach that we use the hydrogels at first and puts the skeletal muscle cells in the hydrogels. And then when we make a pattern of the hydrogels by using the soft lithography method by using the PDMS, so we can actually make a very small, channel-like structures. And within the channel-like structures, the cell can grow and migrate within the hydrogels and fuse each other and make a muscle fiber like this. And those kind of muscle fibers eventually can align the single directions so that once we apply the voltage or electric voltage, they make make a muscle contractions. Using these kind of structures in an array like this, so we can make a two-dimensional sheet, but having the muscle cells within the small channels. And by stacking the two-dimensional sheet onto the Z directions like this, so that we can make a very thick muscle tissues within a very small time. And after stacking the sheet and fix onto the small anchors, and then culture it for about 1 week, so that during the 1 week, the cells can grow and migrate in the small channels and fuse each other and make muscle fibers. So eventually we can actually make muscle fibers aligned to the single directions, but having the very thick tissues within a short time, like 1 week. So this is actually a photo of the muscle tissues. So after culturing the muscles about 1 week, so we can make a millimeter size and millimeter thick of the muscles like this. And if you take a close-up view of the muscles, so we can actually see the very beautiful morphology of the muscles, like muscle fibers is aligned to the single directions, and also having the sarcomere structures like this. And also those kind of muscles have the very unique protein like alpha actinins or myogenins, so that we can actually succeed in tissue engineered the very thick muscles within the, in vitro format. So to actuate such kind of tissue engineered muscles in the robot, so we make a small devices by using the photolithography and the stereolithographies. So to put the muscle sheet and make a fiber onto this kind of devices, so after making the device, so we can apply the voltage between the muscles by using the small, electric circuit. And after applying the electric stimulation to the muscle fibers, they make electric, muscle contractions so that we can actually actuate these small devices. So this is actually a movie how it works. After applying the electric motions, so you can actually see the muscles is actually make a contraction motions and pulls the edge of the small film. So those kind of actuation can be used for the biohybrid robot. And by applying this kind of unit, so we actually make a very small robot fingers. And this is very, when we apply the voltage between the muscles one by one, so we can actually control the muscle finger back and forth motions within the very small area. And this is very small, simple demonstrations, but the world first demonstrations where we actually use the skeletal muscle cells, antagonistic skeletal muscle tissue can be used for the robot applications. So not only for the robot applications, we try to apply this technology for the food industries. So as you may know, the cultured meat industry is now growing and very popular, not only in the United States, but also the Europe or Asia and so on. So if we can create the muscle tissue without killing the animals, so it is good for not only for the animals, but also we can contribute to solve the environmental issues or food crisis, the protein crisis within the next 30 years. So this is we call the world first cultured steak, and which is although the size of the muscle tissue is very small, like 1 centimeter cubes, which corresponding to just 1 grams. So, but within the 5 years, we would like to scale up this kind of tissues, not only for the 1 gram to the 100 grams. So in the other future, we are now actually collaborating with Japanese cup noodle companies called Nishins. So that's in the other future, so we can actually eat the cup noodles with the cultured steak. So thinking about the biohybrid devices, so we can actually think about several kind of the biohybrid devices, but we can actually categorize such kind of devices into 4 groups. So that is the biohybrid sensors and biohybrid reactors, actuators, and processes. So biohybrid sensors, as you can easily imagine, so we can actually detect a very tiny molecule at the single molecular level, like a dog nose or the insect sensors. So biohybrid reactors that can mimic the biological functions like in a body. So when we reconstruct the 3D tissue on a very small chips, so this kind of chip can be used for the drug testing without using the animals or without having the human human experiment. So that can work for the microphysiological systems or organ-on-a-chip applications. So if we can mimic the whole biological reactions in a small chips, so this kind of device can also be used for the artificial organs. So that's this biohybrid reactor can be used for the cell therapy application as well. So the biohybrid actuators, so as I mentioned, it can be used for the robot application for the low energy motions or self-healing actuators and so on. And also it can be used for the cultured steak. And for the biohybrid processes, this is kind of the far future technologies. But if you think about the neural engineering research fields, the many researchers already use the living neurons and make a neural circuit by using the living cells. So if you can actually create the learning networks or memory network by using the living neurons, it can be used for the actual devices by using the low energy processor for the low energy processes. So that we are also working on this kind of processes. But for the sake of times, I just want to mention about what we are now doing for the biohybrid sensors. For the biohybrid sensors, we are now focused on using the insect sensors because the insect has a super sensor on its antenna. For example, the bee has the antenna that has many olfactory receptors. The one of the olfactory receptor can detect the addictive drugs or bombs. Or the olfactory receptor in mosquito can detect the human beings. So they can easily detect the human smell, human sweat, and attach to the surface of the skin and get the blood, as you may understand. So using the mosquito sensors, so we try to apply this kind of sensors for detecting the human beings in disasters. Because in Japan, we often have a huge disaster like a tsunami, earthquake, or typhoon, and so on. And finding a human being in natural disaster is very challenging and very big issues. So once the natural disaster happens, the dogs can work very much. They can easily find the human beings, but the dogs can only concentrate only a 1 hour. So within 1 hour, so that they cannot work for 24 hours. But if you can create the dog nose-like sensors, so it can use for 24 hours in a robot. So that's why we are now focusing on making the, dog nose-like sensors in a robot. But using the dog nose, it is a little bit complicated. So that's why we are now focused on using the sensor in mosquito. So because as I said, the mosquito can easily find the human beings. So what we did is to identify the mosquitoes' olfactory receptors that can identify the human beings. So we actually get the DNA and express the super, protein sensors on its antenna and puts the artificial lipid bilayers. And once the human odors attach to the olfactory receptor of mosquito, they can actually change its conformations and make a small pores onto the lipid bilayers. And after applying, so when we apply the voltage between the two membranes, so we can actually detect the ionic current through the membrane proteins. So if you can detect such kind of current on the sensors like this, so we can actually, detect the presence of the human odors within a very short time. And if you put the sensor signal to the motors, the motor can move back and forth so that we can actually use these sensors for robot. To get the, to force the such kind of sensors, so here is the close-up view of our sensors. So where we have the small chambers contact each other, which we call the double well microchambers. And in between the chambers, we form the artificial lipid bilayer that becomes the platform of the membrane proteins. To form the lipid bilayer on the chip, so we actually use the droplet contact method. So where we developed about 15 years ago, which we call the droplet interface bilayer. So when we put the oil with a lipid, and if we put the water droplets here and make a contact each other, so that we can actually make the lipid bilayer within the very short times and very reproducibly, because the interface between the water and the oil, So we can easily form the lipid monolayers like this. So this is actually a movie how it form of the lipid bilayers. So after making the double well chambers, the size is about the 2 millimeter in length, this size. And when we put the oil with a lipid like this, and puts the water phase here and here. So the two kind of droplets, once the two kind of droplets contact each other, so we can form the lipid bilayer within a very short time, like within 2 minutes. So we actually find that this membrane is actually the lipid bilayer by putting the membrane proteins and I get the signals. So this kind of method is very easy and very simple and reproducible. We can actually make the array of the lipid bilayer like this. And when we put the membrane protein model like alpha hemolysin into the lipid bilayer, so we can easily get the picoampere signals coming from the alpha hemolysis like this. And to use this kind of systems, we try to reconstruct the olfactory receptor of insect. So after making the small chambers like this, and puts the electrode here and here, and puts oil and water droplets here and here, but at the interface between the droplets, so we can form the lipid bilayers like this, as I mentioned. And when we make a close-up view of the lipid bilayer like this, and to form the olfactory receptors, we actually purify the membrane proteins and make a proteoliposomes. So this is a kind of olfactory receptors and make a reconstitute the small vesicles. And after putting the proteoliposomes onto the planar lipid bilayer, they fuse each other and reconstruct the olfactory receptor like this. And after putting the electrodes here and here, so that's, and if we put the protein ligands, like human smell, so that they can, olfactory receptor can move on actuate open and closed state, so that we can get the open closed signals like this. This is the kind of the signals coming from the single membrane proteins. So by using this kind of sensors, so we try to detect the human odors. So if we don't put the human odors, so we don't have any signals like this. But if we put the human odors to our sensors, so we can actually observe the open close signals coming from the olfactory receptors. So that's the so that we can succeed in detect the olfactory human sweat by using the mosquito sensors. So this sensor is very nice for as the selectivities. So if you put the very similar molecules to the sensors, we don't have any reactions like this. But if you put human odors like octanols, so they can actually react very high levels. So for the demonstration to detect the human odors by using the robot, so we make this kind of sensors having the lipid bilayer reconstituting the olfactory receptor of mosquito. And by applying the human sweat absorbed in the paper, they actually detect the human odors and actually move forward. So we succeed in detecting the human odor by using the robot. So this is only a simple demonstration by using the detection of the human odors. But if you change the type of the olfactory receptor of insects, so we can actually detect a wide variety of the odorant molecules, like addictive drugs or bombs or other type of the food odorants or cosmetic materials and so on. But if we can detect the, odor coming from the human beings, for example, if we can detect the tiny molecule coming from our mouse, I mean breasts. So we can actually use such kind of sensor for the healthcare application, like cancer detections or diabetes detections. For the demonstration of the detection of the human breast, so we try to use the mosquito sensors as well, because we found the mosquito also have the olfactory receptor for the cancer markers. I mean the liver cancers. So to use the mosquito sensors and put the our sensors, so one of our researchers tried to detect his human breast by using the mosquito sensors. So this is actually sensors having the lipid bilayer with the olfactory receptors like this. And these bags can correct the human breast like this. And of course, he is a very healthy person. So he doesn't have any cancer marker on his breast. But after correcting his breast, he puts the small amount of the cancer marker onto the bag like this, and then put into the aspirators like this. So when the breast is collected and introduced to the sensors like this, actually we have the 16 sensors on the box. But one of the sensors can detect the cancer molecules within the 10 minutes afterward, like this. So this kind of detection is very important because as you may know, the breast has a large variety of the molecules. There are a lot of noisy molecules, but we can actually detect only a single type of molecules within a very short amount of the concentrations. So this is actually, data coming out from the sensors. So without the cancer marker, we don't have any signals like this. But if we have the cancer marker, only a 0.5 ppb, which is a very, very small amount of the concentrations, you cannot detect, even if you use the gas chromatography without the extraction operations. So that so we can detect 0.5 ppb levels or 5 ppb levels by using the mosquito sensors. So those kind of the method can be used for the correcting the breast and the detection of the cancers within the very short times with very small devices. So that the biohybrid sensors work for the highly sensitive and very small reproducible healthcare application, we believe. So with that, I'd like to summarize my talk. And we actually focus on making the biohybrid devices. And those kind of devices can be categorized into 4 groups, like sensors, reactors, actuators, and processors. And for now, so I only introduce our work for the biohybrid sensors, but we actually working for the other type of the devices like biohybrid reactors, or for the organ-on-a-chip application or cell therapy applications, and also the biohybrid processors and actuators and so on. And in the other future, we would like to combine all the 4 types of the sensors into a single systems where we can actually make a biohybrid robot that can mimic the human beings with highly, high biological functions in the robot. With that, I would like to thank all the group members and the financial support. Thank you very much for your kind attention.
Thank you very much. If you have any question from audience. Can take a look at the virtual session. There is no questions. So thank you very much, Professor. And I'd like to close this session. And sorry, I missed it. Sorry.
Shoji, it's Kit Parker from Harvard. It's good to see you. Glad to see that you survived the pandemic. I hope to see you again in person sometime soon. I want to congratulate you on the work you've been doing on tissue-engineered meat. And I wanted to ask a question because I've been dabbling a little bit in this myself. When you build this with skeletal muscle, are you able to build fat into your tissues so that you can get that kind of flavoring? Thank you.
Yeah, we are now working on. So of course, we actually try to make not only for the muscles, but also the fat tissues. So we actually get the adipose-derived stem cells from the cow and grow into the tissues, and then put it into the muscle tissues afterwards. So that it will be very yummy in the near future.